I plan for this afternoon to talk about two different steps. One is the alignment, and one is the quantification. And I think more importantly, and I don't know if that will reflect in the number of slides, but maybe that's where we also need to spend some time, is to discuss about the quality control after the alignment. Possibly we'll get back to the other computer and see the different parameters that are accessible for TOPHAT, so we can discuss all of them and see which ones are the best for your own purpose. I need to mention a few other analyses that are possible with RNA-Seq as well. So, okay, so here is the mapping or the alignment. So maybe before I say that, here it applies to organisms where we have a reference genome, and that's the case for all of your problems. It's also possible to do RNA-Seq without having a reference, without knowing the genome or the transcriptome. And in that case, you won't be able to align your reads, but you'll have to assemble them into a novel transcript and then see how they relate, and possibly by mapping to a closely related species, how they, if they are new transcript or if they are duplication of transcript or things like that. But that doesn't apply here, and so in all of your projects here, the first step after sequencing and looking at the FASTQ files is to align the reads. And for that, we need a reference sequence that is good or not so good. But anyway, the problem is to match millions of reads that are, let's say, about 100 characters to a reference sequence that contains 1 billion of characters. Okay? And so if you do that by just looking for one sequence, it really takes a very, very long time. And so when this kind of data became available, there was a lot of effort to find the best possible way to do it. The way that was proposed, mostly by Hangley and Richard Durbin here in that paper, but also by the group of Steven Salzberg, is to use this thing called the Burrows-Wheeler transformation that was proposed in the field of data compression. And I'm not going to describe in details this transformation or even this algorithm, but what you need to know is that it requires an index. So we take the reference sequence, we transform that into an index, and then we map the reads to the index. And what it allows to do is to do it really fast, find all possible match of a read to the index, and also allow for some mismatch. Okay? And the first implementation were here BWA by Hangley and Richard Durbin, and that they were part of the Southern Genome Project, and Bowtie. And Bowtie was Steven Salzberg was the Ph.D. advisor of Mike Schatz, who's going to talk later today. And so he will tell you that the way they call it Bowtie because they took a dictionary and they look for BW, I think they look for BWT in that dictionary before Burrows-Wheeler transformation. And the first hit that they found was Bowtie, so that's how they call their algorithm. So both BWA and Bowtie, they're very widely used by everybody in genomics to align reads to the whole genome or whole genome or whole exome sequencing. I would say that for those two applications, maybe BWA is the most widely used. And so they allow for mismatch, and they have multiple options to control the output. So you saw for TOPHAT all the parameters and the options that we have. And those algorithms also have a lot of parameters. The advantage of Bowtie is that it can return all possible match for a read. And to the best of my knowledge, BWA is not capable of doing that. In the case of multi-mapper, so when one read mapped to multiple locations in the genome, BWA will flag that read in the output and return one location at random. Okay? Whereas Bowtie, with the appropriate parameters, can return all the different locations in the genome. And the new version of BWA, so those algorithms were proposed, I think, in 2009 or 2010. And the new version now, they also allow for small gaps in the alignment. Okay? But both of them are used for DNA sequencing, okay, for linear mapping of one fragment to the genome, okay. We take a fragment out of the genome and we map and we want to find that location, okay. The way they report the reads is in the SAM format, and I'll talk about that. So when I say that we take a fragment out of the genome and we want to find the location, that's to make a difference with the RNA-Seq, where the transcript there may result from splicing, and so the mapping strategy has to be a bit different, right? We need to make sure that we can, the fragment that is coming from a gene, from a transcript, can be mapped to the genomic location, allowing for large introns in between, large intron in the middle, right? So there is two strategies. Either we map to the transcriptome, so we make the index that we have to make, we make it out of the transcriptome. So take all the different isoforms in a FASTA file, okay, all the different sequence of the isoforms, make an index of that, map the reads to that index, and that you can use BWA or bowtie, so you won't have an intron. But you need to make sure that you will record all the locations, okay? So if you have 50 isoforms from the same gene, a read from that gene might map to all of the isoform, okay? The most common strategy is to map to the genome, where you need to use what we call a splice-aware aligner, so an aligner that's really developed for RNA-Seq. And TOPHAT was one of the first, and clearly by far the most popular aligner for RNA-Seq data. And so, as you can guess, TOPHAT was developed by the same people that developed bowtie, okay? So that was even Steven Salzberg group, and last year they proposed TOPHAT 2, which is an improved version. So it's built on top of bowtie. And so basically what it does is to split the reads into pieces and map them to the genome with bowtie. And then for the reads that don't map them, that don't map to the genome, and I'll probably try to describe that. So you have a 100 nucleotide or 100 nucleotide read. You would split it into pieces, and if you can map that location to the genome, so yeah, let's say you can map that location to the genome. You can map that location to the genome, okay? But this piece here won't map directly to the genome. You would try to extend this one, okay, up to here, and extend that one up to here. Okay, and here you have a splice junction. Does that make sense? Okay.
One read that you're lining up?
So that's one read here. Okay, and that's the genomic location. Okay, so
It has an algorithm to keep doing that if your smaller piece of read is in the middle.
so that's what we have here. So you split the read into pieces, right, and then you map to the genome. Okay, so that piece here will map perfectly to that location in the genome. The end of the read will map perfectly to the genome, but that one won't map perfectly to the genome. Okay, there is no location that match for this one. You will make it match by extending the reads here up to a certain location, and after that it stops mapping. Okay, so the part that is here, they won't map here. Okay, it will only start to map here. Okay, and so that one you get by extending the end portion.
What if you've got lots of splice? I don't know.
Well, so
Splice segments.
right, but so for
some of these things can have 200 different.
Right, but PacBio are multiple kilobase.
Right.
So the PacBio can have multiple kilobase reads.
Right.
So they can have multiple splice.
What I'm saying is that there may be a lot of splice variants that we don't know. Maybe.
Well, so here, but here in the strategy that you described, you don't know the splice variant. You don't know the splice junction.
Right. But you're showing that only one of those three
Yes.
is in the middle of a splice.
Yeah.
What if all three of them?
Well, so that would require so let me try to describe it. The same read here. If you have a splice junction that's here, one that's here, and one that's here, first you're not lucky, but that would require a genes that's like this, then like this. So that requires exons that are like 2 successive exons that are 50 nucleotide, like that are not even like 30 nucleotide long. You see what I mean? So they're really, really small here. That's unlikely to be.
They're so small that the probability.
Right. And so, but that's probably taken into account by the algorithm. Here I describe with 3 pieces. You can actually do with more than that, right? And so what it would do is, if he finds it would split in 3. If he finds 2 locations that are fairly, say less than 10 kb apart, then that's a good alignment, and he will try his best to find where this maps. Okay, where does this fit? And maybe here there is actually, this part has a mini exons in the middle or something like that. Okay, or maybe there are some mismatch here that makes it hard to find. Okay, but there is multiple strategy for that. Okay, but the idea is really that out of 100 nucleotide fragment, and again, that algorithm was developed when the reads were probably 50 nucleotide. Out of 100 nucleotide fragment, the likelihood that there are 2 splicing junction is actually quite low. Okay, and even if it's low, you can take it into account. And out of all the reads that fail this strategy, find another strategy that's a little bit more computationally intensive, but that will apply on a much smaller number of reads. Does that make sense?
Yes.
Cool. So yeah, so break the reads into pieces, map them to the genome, and then extend the possible splice junction. One option for TOPHAT is to give as a parameter an annotation file. When you have a good annotation, you can give it as an argument, and that will guide the alignment. Okay. So to get back to my example, here you might have that might be a small fragment here. Okay, maybe a few nucleotide, and so there could actually be multiple possibilities to map this. Okay, or yeah, let's say. What do I want to say? So out of this one, you can have this or no. So you might have 2 different locations for the splice junction. Okay, when you extend your read, maybe this part will map, and then that part will map, and you'll have a junction like this, or you'd have that one, and both are equally good. You see what I mean? So, yeah, I'm sorry. It's
extensions made to, depending on the presence of consensus splice.
No, so no. In what I described before, no. When we give it an annotation file, then yes. It will, if one of those 2 choices correspond to an annotation site, it would probably favor the one that is already known. Okay, so that's kind of based on the annotation, not so much on the canonical splicing site. But for equally likely alignments, it would favor the one that correspond to a canonical site. Okay, so it knows.
I think the canonical or the consensus.
Yeah. Okay.
So is there
So the warning for TOPHAT or for bowtie is that it's clearly developed for mammalians genome, and there are a few parameters that need to be changed for other ones. I don't think you can change the knowledge of the splicing canonical sites or consensus sites. But I don't think it's possible. But again, if you have an annotation, you can give it as a parameter, and that will help. That will guide the alignment to this annotation. With the issue that you bias towards known junction, and so you have a little bit less power to discover novel splicing junction, right? So that's a trade-off that you have to find. The new what is it? So yeah, when you give it an annotation file, it will actually extract the transcript from that annotation. So from the coordinate of the genes in the annotation, it would extract the transcript, build an index, map to the transcriptome the way I just described before, and then to the genome. So out of everything that doesn't map to the transcriptome, try to map it to the genome with the strategy I described here.
So you're making are we like downloading a GTF file or?
Yeah, so it's optional. It's optional. If you have one, you can provide it. I don't know if it's accessible in DNA Subway, but we'll look at it.
So in Subway, I guess it automatically pushes the GTF with it.
Yeah.
Again,
okay.
This is a feature request. When there are multiple versions or histories, you can in the future support if you want to go back. And certainly in the discovery environment, you can choose
okay.
and mix and match.
Okay.
So when you say extract transcript, sorry, I'm just trying to get it.
So the GTF format is the set of coordinates. Right. Okay. So you have a reference sequence, a set of coordinates, so you can extract the sequence corresponding to the genes, to the transcript. Okay, that's what I mean. That corresponds to your known transcript, and out of which you will create an index.
So you're building an index from the GTF.
Yes, well, from the FASTA file and the GTF. You create a transcriptome.fasta, but you don't do that. The algorithm does it, right? You don't have to worry. So using annotation that improve accuracy mostly around splicing junction, it bias in favor of known transcript, but there's less power to detect novel transcript or novel junctions. And so that's something you have to be careful if you have incomplete annotation or not so great annotation. The question of multi-mappers. So for each read, it comes from one location, but there might be other locations in the genome that match equally well. Okay, and so for exome sequencing or for genome sequencing, we usually don't keep the different locations. For RNA-Seq, we tend to keep the multi-mappers. Now, it's not a bad idea, or you can record all the locations, but you might have some reads, and usually they are not very informative. They are low quality reads that may map to multiple locations, and especially the shorter the reads. So when you do the trimming, if you end up with a read of 20 nucleotide, that's actually very common. It can map to thousands of locations in the genome. Okay, and so in your output file, you'll have 1,000 lines, okay? And none of them are informative. You probably won't do anything with this read and with that information. So it's not a bad idea to put a limit. And so there is a parameter in TOPHAT to set up the number of locations that you want to record per read, the maximum number of locations that you want to record, okay?
Can I ask a question about this?
Yeah.
So if you make a limit of, let's say, 10 or whatever that limit is, does it mean that anything that maps to more than 10 will be thrown away, or does it mean that
so
all the first 10
again, that's the two possibilities. Both are possible in TOPHAT. So you can say, give me all the reads up to 10 location, all the location up to 10 location, or give me only the reads that map to less than 10 location, and don't output the reads that map to more than 10 location at all. Okay.
So you sort of had your choice.
Yes, in TOPHAT, you have the choice. I briefly looked at the output, the parameters for DNA subway. I think there is a whole lot of choice also, and we'll go through them together if you want. Yeah.
This sounds a little like a deduplication.
Sorry?
It sounds a little akin to the deduplication issue where you start ignoring these multiple reads.
Yeah. So.
This is not multiple reads.
Yeah.
This is one read going to like 5 different places. Imagine a
Yeah.
composable element. So when you pick
Right, that's fine, but you know, somehow or another you're only sticking it to one, but it actually corresponds to this other site, which maybe I'm.
So it's two different things. One is the exact same reads coming out in the FASTQ file, and one is one read multiple location in the alignment file. Okay.
I'm sorry, I can't hear you.
So it's a different issue than the duplication that we discussed before. Before it was the exact same reads present multiple times in the FASTQ file. Here we're talking about one single read present multiple times in the alignment. Okay, and they're similar in the way that they will affect the gene expression, the estimation of the gene expression, which is the ultimate goal for that. Okay, I need to.
For each gene in the gene family, they would count 10 times. I mean, in a sense.
So one read, if you don't pay too much attention, one read, if it maps to 10 different locations, you would count. You sequence it once,
right,
and you will assign it to each of the genes.
So
Okay,
count it 10 times.
that would be counted 10 times. Okay, so that's where you have to, when you do the quantification, take into account that it maps to other location, and one trivial way is to count it 1/10. Okay, one out of 9. Okay, and that's what CUFFLINKS is going to do, for instance. Okay. So I need to. I don't know if that's been described to you, this SAM and BAM format, and that's extremely useful. So that's the file that results from the alignment, and that's how we record all the different alignments. So it's composed. So first, it was the format was proposed by the Southern Genome Consortium, and it's composed of 2 parts. One is the header and describes the source of the data, the reference sequence, the version, the method of alignment, and so on and so forth. And the second part is the alignments by itself, describe the reads, the location, and the nature of the alignment. Here is an example of a header. So I'll describe this first line after. So that's the command line that can be used to visualize the first line of this file. Okay. So the file here is called sam.bam, and I'll describe then the difference between SAM and BAM. But you see here that you have a description of the reference sequence with the chromosomes and the number of nucleotide in the chromosome for that version of the reference sequence. You also have so that's called the read group that summarizes some information. So the platform was Illumina, the instrument was a HiSeq 2000. That's the sample, that's the Genome Center here. That's the command line that can be used. And here, I didn't include it because it's very long, but that's the entire command line with all the different parameters. So this is indicated in the header of the file. And that's an alignment file, okay? So that's just the first 5 lines, and there are probably 10 million of them. So you can all read that, right?
Yes.
Okay, so.
It's the same, it's 4 lines again.
No. So that's just one line, it just doesn't fit on the screen. Okay? So here is one line. Okay? And you had any question? Somebody had a question? No?
Can you go over what you, I'm sorry.
Yes, I can. So here maybe you can recognize it. It's the header, and that's exactly the same as we had in the FASTQ file. Okay? So that's just exactly the same information. Here is what we call the flag, and so the flag is described in the specification of the SAM format, and it will tell you, so it's a bitwise, so this basically adds up, and so 99 is a sum of different flags, and so it tells you if the read is mapped or unmapped. So if the flag is 4, it means that the read is non-mapped, okay? So the SAM can contain the mapped reads as well as the unmapped reads. It will tell you if in which orientation is the read, okay, with respect to the reference sequence. It will tell you if the pair is mapped, okay, so when you do paired end sequencing. It will tell you if it's a secondary alignment, which means that there are other locations for the same read, okay? And so I indicate at the end there is a website which explains flag, okay? So every time you have a read, you can take the flag and it will give you all the explanation for that flag. Here are the location of that alignment, the chromosome, and the leftmost position of the alignment. This is the mapping quality. And so the mapping quality depends on the aligner. So that's widely used, again, for exome and genome and by the aligner called BWA. In RNA-Seq, it's usually not very informative. I just think the aligners, both TOPHAT or other competitor, they don't do a great job at calculating this quantity. This is called a cigar string. So cigar is, that's kind of what we can say as the nature of the alignment, okay? So it says the cigar operation are given by the following table. So the M stands for match, okay, the alignment match. The I stands for an insertion, D for deletion, N for skip region from the reference, S is soft clipping. So we don't have soft clipping here, but I'll explain. And yeah, we don't use those ones so much. So here, 23M. So we have, out of this sequence, which I believe is 50 nucleotide, the first 23 match the genome. And then we have 1I, so we have an insertion of 1. And then 26M, so 26 match, okay? So on this thing, the first 23 are mapping the sequence. And then there is one nucleotide that is not in the reference sequence, and the rest of the 26 remaining sequence base are matching the genome. Okay, so that's not the most complicated cigar string that I could have found, but essentially that's a way to describe the alignment. Okay, to describe which are the locations that match, where do you have an insert or mismatch or deletion, and soft clipping is when you have extremities that are not matched. So say you have a 50 nucleotide read, and it's going to match on the 35 nucleotide. The last 5 won't be called mismatch, they will be soft clip. So those ones won't match, okay? They will not match the reference sequence, but they will still be part of the read, so they will still be reported here.
So if I was to run my SRA files with all the what do you call them?
The
adapters, then I would have a lot of
some, but most probably a lot of those reads won't map the genome. That's what you would expect. So if half of the read,
the first part of the read would.
Yeah, the first part, but so usually the soft clipping is it's not for half of the read, right?
Yeah.
But that kind of depends on the aligner as well, so that there could be differences here. But the way you do the alignment is really by finding the best possible location and then scoring that. And so going over the entire alignment and making a score that gives a positive score for match and some penalty for mismatch or for an indel. And so when you have too many of those penalties, you are below a threshold and you don't match, or you don't report that alignment. Okay?
It's a little bit like last time.
Yeah. Yeah. Very much so. I mean, it's an alignment strategy. Yeah. if you ever saw a cigar string for a PacBio reads, that's very informative. And that's like, you know, take like half of this screen, and because it's very long, and also because there is much less match than for Illumina. So there is a lot of, you know, insertion, deletion, mismatch, and so that makes some interesting results. Let's continue through this file. So here, so that's the chromosome of the paired read. Okay. So here, in that case, it's on the same chromosome. The location, okay, so we see that it's pretty close to the read that we're currently looking at, and the template length. So so from the leftmost, from that location here to the end of that location here, okay. And so you have to be careful because here we're talking about genomic coordinate, and that's different from the fragment size, which can be on the transcriptome coordinate. Okay, they're coming from the transcriptome. So you can have a fragment of a few 100 nucleotide on which the genomic coordinates are 10 kb apart. Okay, because there is an intron in between. Yep.
Is this all one line?
Yeah, that's all one line. That's all one line. One line per alignment. Okay. I just wrapped to fit on the screen.
Right. Okay.
Okay. Here is the sequence of the read, the quality score of that read, and oops, sorry. And then there is a bunch of optional fields. One of them is the read group, and that's some of the same information as we had in the header. And then there is, I can't read that. So there is additional information on the number of mismatch. I guess it's, I think it's here. So I think this one has no mismatch, or here maybe. But the entire specification for the SAM format are available, and I have the website at the end. And the advantage of that format, so you know, you could imagine many different ways to report alignments. The advantage of having all the aligners using the same format is that many tools are developed for that format. It's clearly a you know, community standard. It comes with a software called SAMtool and a C library. So there, you know, it's possible to have a lot of tools that can read into those files very efficiently. Also, the key advantage of SAM is that it can be compressed into the BAM file, so the binary version of SAM. And those BAM files can be processed directly by SAMtools. And they can be sorted, can be indexed, and they can be accessed randomly. So that means you can have a multi-gigabyte file and access directly to the alignments corresponding to a specific genomic location. So you do SAMtool view your file.bam, and the coordinate, chromosome 1, semicolon left to right position, and you will have all the reads corresponding to that location. And you can do that without having to read the entire file. Okay? Remember, the file is several gigabytes. If you have to read all through it, it will take a very, very long time to read and to capture the one regions you're interested in. The advantage of BAM is that you can go directly to the genomic location you're interested in. Okay? And that's a absolutely key advantage. The specification of the tools are here at this samtool.sourceforge.net, and the file that explains the flag and that also contains another library that we will talk about a little bit is this website here. What we can do when we can access to a specific genomic location is to visualize all the reads. So for instance, this browser here is called IGV from the Broad Institute. And so here, you know, you specify the genomic coordinate, and here that's where the genomic coordinate are. They would look into the BAM and collect all these alignments and report them on the browser. So we can go through it. So here it's the organism, the chromosome, the location is here, There's a number of buttons here to navigate in this software. You have this ideogram, which represents all the bands on the chromosome.
Just dragging one BAM file into IGV?
Yes, that's just one BAM file into IGV, okay? And that's how RNA-Seq are represented into IGV, okay? You have, so here are the different, the genomic coordinate, the coverage scale that shows you that, there is a lot of reads corresponding here at that location, that location here, and they turn out to correspond exactly to the different exons of this read. And you have all the reads. So the part that match the genome are here in red, and the blue lines here are the junctions between the part of the read. Okay, so so the read part match partly here, and the other part match here.
So each line is one read.
Each line is one, or actually, well, so here on the first line there is multiple reads. If we zoom in, we'll see that here there is probably multiple reads. There's more than one read per line. Okay. So if you see here, here for instance, there are probably 2 reads. Yeah.
But it would be it's probably from pair
yeah. So
it's actually read 1 and read 2.
it's probably read 1 and read 2. When you zoom in, you really can see the details of that.
What I mean is that if 2 pink squares or whatever you call them, boxes, are connected with the blue line,
Yep.
it means
It's
that
the same read that match to the 2 different locations.
Right. So
Okay.
a splice was detected by the software.
Well, a gap,
Yeah.
and when you have a lot of gaps and that have the same location, that's probably a splice junction. And so again, I really encourage you to look at the file into IGV, and that is very informative. That can tell you a lot of things about the quality of your data, and sometimes that you can really make discovery by looking at those files. If you have, say, 2 different locations and you want to see alternative splicing, you really can see it from IGV. You can load 2 files here. Okay, so you have a separation here. You have 2 files, and you will see that one exon is used in one condition and not in the other one. That's the kind of thing you can see with this alignment.
I'm sorry, what's VE?
Here, that's positive strand and negative strand. So here it's probably a stranded protocol, okay? So on which the coloring depends on the orientation of the reads. The red are probably in the same orientation as the gene, and the blue here are corresponding to a transcript in the other orientation. Does that make sense?
So that, okay.
Yeah.
So that's positive, negative strand on GeoMid DNA?
Yes. Yes. So that was kind of the end of the part on the alignment. Okay. So now we have an alignment file that's record all the location of the reads, and we'll talk after that part. We'll talk about the quality control we can do on this. What I want to describe here, and that will be expanded by a Molly Hamill presentation on next week, is the quantification. And so usually you don't want to stop at the alignment. It's crucial to do the alignment, but you will do something else after. And so what you want is to have a good estimate of the gene expression. Okay. Now, when we say gene expression, we have to see what exactly it means. Okay. And that's what we're going to discuss a little bit. Here is an example of P53 in the human genome. You have 1 gene, and you have 15 annotated transcripts. Okay? So when we talk about gene expression, do we talk about the expression of like the most common isoform, or we talk about the different transcripts? Okay? So I'll describe a gene as this one, or the region that codes for this protein, and then I will describe alternatively transcripts or isoform into the different form. Okay, so here we have 1 gene and 15 isoforms or transcripts. Is that okay for? so the tool that kind of goes with TOPHAT, and that again is very, very widely used, is called CUFFLINKS. You can see the theme here. And so based on the alignment, it will try two things. It will try to assemble transcripts, including new transcripts, and try to discover new isoforms. And it will quantify the transcripts, okay? And so it was proposed by this paper in 2010, again by Steven Salzberg and a few other investigators. And the first author is the same as for TOPHAT. So here is the main figure of their paper. So for the assembly, which is that part here, the goal is to try to find the minimal number of paths on a graph to fully represent the alignment. And I mean, I guess that will be described better by Mike Schatz later today. But you have an alignment, and you don't know any. So if you don't know anything, about the transcriptome, you'll try to determine how many isoforms, how many transcripts are necessary to explain the alignment that I observe. Okay, and so you will connect the different reads with their splice junctions, and that will create a graph like this, and you will determine that there are clearly three ways to navigate within that graph, and that correspond to three different isoforms, which have some portion in common and some portion that are different. Okay. The other thing that CUFFLINKS is trying to do is to estimate the abundance of the transcript, and so here the idea is really what's the based on the alignment, what's the most likely abundance for each of the transcripts. So for that, you need to know the transcript. So either you discover the transcript with the first step and then you quantify them, or you take a known annotation as a parameter and you try to quantify them. Okay? So in CUFFLINKS, you can skip the assembly part. Okay? You don't have necessarily to go through the assembly of novel transcript. You can skip that and focus on the quantification methods.
So it will give you the quantification. Would that be based on, like going back to what you were saying earlier, to every transcript from one gene or individual? So
it mostly works at the transcript level. Okay. So we'll try to quantify every isoform. And so again, you can tell him to determine how many isoforms there are to explain the alignment, or to restrict on the set of known isoforms. Okay? And that set of known isoforms will depend on the annotation file you will provide. And as you know, annotation can be very stringent or very exhaustive. Okay? So in human, if I work with GenCode or with RNA-Seq, it's a different set of isoforms. So in order to summarize the quantification, we need to talk a little bit about the unit, like what do we measure and how do we represent this estimation. So one thing to really realize when we do sequencing experiment is that everything we measure is relative, okay? Everything is based on X number of reads in the library, Y mapped to gene A and Z mapped to gene B, okay? But if you change X, if you have twice the number of reads, both Y and Z will change, okay? What you measure is not an absolute level of expression. It's always relative to your experiment, relative to the number of reads that are present in your experiments. And so what it means is that the expression level that you measure is relative to the expression of the other genes in the same experiment. Okay? Does that make sense? Is that okay? So really, if you change the depth of the library, the number of total reads in your library, you will change the expression of the genes. And so we need to account for the depth of the library. When we want to compare two samples, we need to make sure that we took into account the total number of reads in the library. Now, when gene A and gene B have exactly the same number of reads, do they have the same level of expression? Well, the answer is no, because, well, the answer is yes if they have the same size. Okay? If they have the same size and the same number of reads, then it's safe to say that they have the same level of expression. But if they don't have the same length, the same size, then the level of expression is probably not the same, because for a longer gene, it will receive more reads than for shorter reads, shorter gene.
You should say gene here, you should say transcript.
Yeah.
Because a gene could be the same size, but have very different transcript size. And since this is RNA-Seq.
Sure, sure. Yep, good point. Yeah, no, exactly. So exactly, and CUFFLINKS really work at the transcript level. So let's put transcript here, no, but it's exactly that. Very good. But I mean, so you all know that, like, even for two genes who have the same level of expression, there might be different transcript. And so we need to account for the difference in the gene lengths, okay? And so what CUFFLINKS proposed was this measure called FPKM that you might have heard of. It's also in single read experiment called RPKM for reads, but in paired end, it's called FPKM for fragment per kilobase per millions of reads, okay? So FPKM, so what it actually really means, so that's say kilobase here, but what they mean is not so it's not the annotated gene size, gene length, but what they call the effective gene length. And by effective, they mean the number of possible start sites in a transcript, which depends on the fragment size, okay? So if you want to, I can describe that. Okay, and so that's the fragment. So we fragmented the transcript into those things, and we're going to sequence the, I don't have another color, but that's fine. What we're going to sequence is here. Oh, yeah, okay, they're working too. Okay, cool. Wait. Oh. Yeah, it's working. Okay, so what we're going to sequence is those blue bars here. And so what we consider the effective size is that size here. Okay? Because that's the last start site that you can have for a fragment. Okay? So it's basically the length of the annotated length of the transcript minus the so what they consider is the effective gene size. It's the length, the entire length of the so it's the number of possible start sites for a fragment. Okay, and so here I describe here the most 3' end fragment. Okay, which starts at that location. So that's the rightmost start position. Okay, that's the start position for a fragment. That's the most on the 3' end. Okay? You can't have a fragment that starts here. Oh. Okay? Because your transcripts finish here. Okay?
Oh, I see. That end of the
Is that because that would make the fragment too small?
Well, because you have this transcript, you fragment it, and then you sequence.
Right.
Okay? So you can't have a fragment that's here.
Yeah.
If you fragment it, the fragments are that size,
Yeah.
and so that has to be the last position. Okay?
So that depends then very initially of what was the average size of your fragment.
Exactly.
when you made the
Exactly. So the first step in CUFFLINK is to look at the BAM file, the alignment file, and to estimate the fragment size. Okay? And from the fragment size, it estimates the effective sites per transcript. But so that's something to consider, because if you compare two different samples that were sequenced at a very different time with different people and different technology, the fragment size might be very different, right? And so that measure can vary, okay? So that's what they mean by kilobase, is the effective gene size. And by millions of reads, I actually asked them pretty recently, and I believe that what they use is the million of mapped reads and not the millions of sequenced reads, okay? It's not the total number of reads in your library, it's the number of reads that map the genome, okay? And so that might make a difference. So FPKM lets you compare the expression of one transcript between samples, because it accounts for the difference in library depth. But it also lets you compare the expression of two transcripts within the same sample, because it accounts for difference in gene length. Okay? So you have those two normalization that lets you do those two kind of comparison. Okay? That's why that's why it was used. So in addition to estimating transcript expression, CUFFLINKS also output the gene expression, so which kind of more or less is the sum of the expression from the different isoforms. It also comes with a method called CUFFDIFF for differential expression. Okay, so you would provide the set of alignments for one condition, the set of alignment file for the second condition, and it will estimate the isoform expression and perform statistical tests for differential gene expression between the two groups, differential transcript level between the two groups, alternative splicing, and differential usage of transcription start site. So is there some genes where the start site is the start site that is used is different between the two groups? Okay? So it's built on the same approach as CUFFLINKS, but in addition, it does the comparison between two conditions, okay, between two groups, and that's probably what you wanna use in your experiments.
There are many loci, and most, if not most, that have different isoforms, but there are so few for which there has been proven to be or shown to be any functional implications of that. So, when it's, I mean, I've been thinking ahead to the pedagogy. I would lose students going through this quantitation
Yeah.
step.
Yeah.
I think I don't want to shirk responsibilities in trying to convey the nuts and bolts of an RNA-Seq experiment.
So, I mean, here is the CUFFDIFF proposition. Yeah. I actually don't use it, to be very honest. And I mean, I can describe, another way that's actually in a way more simple, that stays at the gene level, where you don't go at the transcript level, and you really try to capture different expression in genes between the two conditions, okay? And for many experiments, that's sufficient. That's what you want. Now, you also, theoretically, with RNA-Seq, you have access to isoform resolution, right? You can look in detail at your alignment and say that, in the two conditions, there are transcripts that, genes where the transcript is not the same. The use of the transcript is not the same. And so it's good that we have tools that, can try to go at that level of resolution and take full advantage of the data that we produced. it wouldn't, I mean, really to work as nice as described here, you need a lot of reads and very high quality reads. And usually it's better if you have good annotation as well. But you can run CUFFLINKS and CUFFDIFF and stay at the gene level and say, you know, CUFFDIFF determines that 50 genes are differentially expressed between these two conditions. It's possible. You can also go into transcript level.
Well,
so.
Again, I'm probably jumping ahead because it seems next week is more focused on the development of pedagogy and curriculum.
Yeah, so next week, and mostly Molly will describe the statistical analysis that is taken care of here, okay? And so yeah.
So so you're saying that it would be good for genes, but not necessarily, if you really want a good idea of how much isoforms differ, this probably CUFFDIFF unless you have like perfect.
So my next slides are different alternatives to this approach, okay? So website, but otherwise it's possible to give a FASTA file and make an index out of that. So you can run any of your organism, as long as you have a reference sequence in FASTA format, you can create an index. What I'm saying is that star index will be much bigger than the TOPHAT index. Okay?
Nicolas,
Yep.
I'm really not sure I understand really what the index is.
So it's a
Sorry, I know you mentioned before
it's a representation of your reference genome in a way that that can be queried for each read. For each read, you try to find the location on the reference genome. That's the mapping. And to do that, you need to represent your reference genome into an index, into a graph format that tells you that, well,
Sorry about it. Okay.
no, it's complex.
So she's doing honeybee,
So you can do it. So
you do it.
So you do it. So you can do it. So TOPHAT or star or bowtie or any of these aligner, they come with a command line that lets you create an index out of a FASTA format, out of a FASTA file. Okay? So input is a FASTA file, reference genome. The output is an index. Okay?
So we actually do that for you in DNA Subway. So
Yeah.
when you choose what organism you're working with, along with loading in the reference FASTA and annotation, we pre-built the index from the FASTA.
But so for information, they don't have to pre-exist. You can do it as long as you have the FASTA file. Okay? So that was star is a very good alternative to TOPHAT. I'm not saying that everybody should use it. I'm just saying that it's important to know that there are alternatives. For CUFFLINKS and CUFFDIFF, it is possible, and I say here sometimes advice, to simply count the number of reads mapped to genes. So you don't go to the isoform resolution. You don't do transcript per transcript. You really only count reads, how many reads map to each intervals, each genomic location corresponding to a gene. And you don't normalize. So neither by depth of sequencing nor by gene length. You just count the number of reads mapping to each gene.
Then what would be the total number of reads or the average number of reads?
The total number of reads mapping to that location. Okay, and then there are rules for what if a read maps partly, is just overlapping a junction or overlapping the extremity of the reads. But there are two popular scripts for that. One is called RNA-Seq. One is feature count. This one, feature count, is much faster, gives similar results. So for the same reason as before, that's the one we use. And typically takes, let's say, 20 minutes to count all the genes in a BAM file. So depends on the size of the BAM files and on the number of genes you want to quantify. But that's a very efficient way to count the number of reads. The advantage of working with reads, raw reads, is that you can input them into those packages for differential expression analysis. Those are R package in this library called Bioconductor. I don't know how familiar you are with R, but they're really developed by a statistician, and it's much more elaborate than CUFFDIFF, and it gives you access to much more sophisticated statistical modeling. So you can specify multiple, like a complex experimental design with 2 conditions and covariates. And so, and I really can disentangle the covariates and find exactly what's different between the 2 conditions. It's really powerful. That's really more than I can describe here. I believe Molly will touch on that and describe the normalization and the statistical tools for some of those packages. And that's.
These are installed for you on an atmosphere image. So when we get to that, and those who really want to do some of the advanced things,
okay.
We have some virtual machines
okay.
that can do that for
cool.
you.
Cool. So yeah, so it gives you access to more complex experimental design. The other alternative that I briefly described before was, instead of mapping to the genome, you and try to assemble and quantify the transcript, which, as we see, is hard. You can make an index out of the transcriptome sequence, okay, the transcript sequence, and map directly there, typically with bow tie, because you need to get all the possible mapping locations for the reads, okay? So that makes you like a very big alignment file, okay, because the header, instead of having all the chromosomes, will suddenly have all the transcript with their size, and then you would have, for each read, all the different locations it can have. And so it can map to 15 different isoforms, okay, if there are 15 isoforms, okay? But then out of that, you can use one of those 2 tools that are quite equivalent, and that will estimate what's the expression of each transcript, okay? So based on the expression, and so I can also describe that briefly. So you'd have, let's say that the isoforms just differ by their UTR lengths, okay? So they are just different size, and so you have the coverage on this transcript will be like this. The coverage on this transcript will be like that, and the coverage on the third one will be like that. Okay? So here, if you have those 3 type of coverage, the most likely one is this one. Okay? So that's probably the isoform that is expressed in your sample. Okay? So here you have a uniform coverage, okay? As well as here, but all the reads that contribute to this coverage here are also present in that isoforms, okay? As well as they are present here, but here you see that you don't have coverage on the extremity, okay? So that's what RSEM or Express is trying to do, is to find out of these coverage plots what is the most likely transcript for these alignments. Okay, so that was the different just alternative strategy. I want to briefly mention quality control. So I don't know if it's going to be brief or not, but we'll see. There's a number of metrics that we need to estimate out of the alignment or out of the quantification. The first one would be the ribosomal contamination. Okay, so as you know, in cells, most cells, ribosomal RNA is highly abundant, and you can have different strategies to try to remove the ribosomal content before preparing the samples. But those strategies are, not perfect, and you will still sequence some ribosomal content. So what we do at the Genome Center, and there are multiple strategies for that, what we do is to map the entire library against a set of ribosomal RNA sequences and count the number of reads that map. But we don't filter out those reads, okay? We'll still map them to the genome, and they probably will map to the ribosomal location, to the location of the ribosomal sequences. But we want to measure, we want to estimate what percentage of the library is corresponding to ribosomal sequences, okay? So that's, we do that by basically an alignment to a short index composed of the ribosomal sequences, okay? The basic number that really are important to look at is what is the number of reads that map to the genome, what are the number of reads that map uniquely to the genome, and how does that correspond to the total number of reads that were sequenced in the library. If you have 50% of the reads that were sequenced that map to the genome, means 50% of them don't map to the genome, right? So either they're bad quality or they're contamination by another organism, okay? And that's something that can happen. Again, if you have a very low percentage of reads that map uniquely, it means most of the reads will map to multiple locations, and there could be reasons for that. If you study microRNA or piRNA or transposable elements, there are good reasons for that. If you study mRNA, you should not have, there are organisms where that would probably be the case, but again, no strict rules, but you have to look at this number. And importantly, you have to look at this, so within the same sample or same project where you have multiple samples, you probably won't expect to have a large difference in the ratio of number of reads mapping the genome to to the total number of reads, right? Except if you have good reasons. The distribution of expression. So typically in RNA-Seq, you have very few genes that are super highly expressed. You have some genes that are mid-level of expression, and you have a lot of genes that are lowly expressed, including some that are not expressed at all, okay? And if you sequence deep enough, you'll have reads that map there, but like just a few reads, and that might just mean that they're not expressed, okay? But that's the typical distribution of expression. If that's basically what I can say. Strandedness. So here I heard go on.
I missed the point that I don't understand how the distribution of expression serves as a measure of quality control.
What you're kind of saying, like this is what you expect.
So if you have, if no genes are highly expressed.
Okay, so there are expectations built into this.
Yeah. When I see a library where no genes are very highly expressed, I kind of feel there is something wrong. Okay, that depends on the cell type. That depends on the tissue. If you do blood RNA-seq, you expect hemoglobin to be highly expressed. Okay, and in most experiments that I saw, you have a few genes that take really the the first 5 genes take as many reads as the genes number 5 to gene number 50. Okay, if you rank them, if you rank the gene by their expression, the sum of gene 1 to gene 5 is probably equivalent to gene 5 to gene 50, which is probably equivalent to gene 50 to gene 1000. You see what I mean? Right. That's the kind of distribution that you expect. And that's not a strict rule again, but that's very frequent, that really the very top of your list will account for a large portion of the library. Okay? Now where exactly does that fit? That kind of depends. But that's a typical distribution of expression that you would expect. Okay? So here, strandedness, your experiments were with stranded protocol, and so you expect most of the read that map to a gene to map in the same orientation as the annotated gene. Okay? And so what you expect 100%, and you usually obtain up to above 98% of the reads that map in the same orientation. Those protocols are quite robust. If you have much less than that, that's something to be worried about. It can be either annotation, so that measure depends on the annotation, but it can also be a problem in your library preparation. And for non-stranded protocol, of course, you expect 50%. You expect reads in both orientation. Okay? And the insert size, so that's those ones here. okay? So the difference between the extremity of read 1 and read 2. And you want the more tight distribution, okay? So you want to have, you want all the fragments to be around the same size, okay? And so you don't want it to be too short in order to avoid overlapping reads. And between conditions or between the group of samples that you want to compare, you want as much as possible to have a similar insert size distribution. The other metrics that we measure, so that's the distribution of GC. That's you had from the FastQC, okay? You can also compute it from the alignment, and so that might be slightly different because that's only the reads that map the genome that would be considered, and so all the long reads of Gs that we saw this morning, they won't map the genome, and so that will change a little bit the distribution of the GC content. The duplication rate, same. We can measure that from the FastQ. We usually do it on the alignment by looking at the location. So when two reads are mapping to the exact same location, for highly expressed genes, that's okay. If you have 50 of them that match exactly the same location, that's the same sequence, that's something to be a little bit worried. And so you can measure this duplication rate, and that will tell you like what's the percentage of the reads that map to the genome that are duplicated. And there is a way to remove those reads if needed. Okay, and again, usually we don't. There might be cases where you want to do it. Percentage of the reads that map to the coding sequence, the UTR, the introns, and the intergenic region. You do poly-A selection, you expect to have mRNA. You expect most of your reads to map to coding sequence and UTR, and few of them to map to introns, and even fewer to map to intergenic region. That's if you have a good annotated genome. If it's not as good, you might have real coding sequence that are considered as intergenic or intronic regions, right? Gene body coverage. So gene body coverage, you check the uniform coverage along the gene, and that you don't have a 5' bias. Those ones are quite rare, although we do see them sometimes. But most importantly, you don't have 3' bias, which are more frequent because of poly-A capture, okay? But so the way to look at that is really by taking the annotated gene, and for each alignment, each mapping location, see on which bin it falls into along the gene, okay? And then you do the average per bin along a gene, and so you have those curves that.
Is there an algorithm for doing it?
Yeah. So all of this, I'm gonna tell that. So there is an algorithm that does this one, this one, this one, this one, and it's part of the Picard library. So I mentioned Picard before, and you had the website. And so it does all this. There is another thing called RNA-CQC or RCQC, and I think I have the website after. The last one is not actually part of those tools, but I suggest to do it is just to do an unsupervised clustering of your gene expression. So you have your count metrics so all the counts or all the RPKM for each sample, and for each gene you have the RPKM, and you do an unsupervised clustering to see if the samples will cluster according to some biological expectation or according to experimental batches, or they won't cluster, or one of them, or they cluster. Say if you have 4 replicates in 2 conditions, and suddenly you have one sample in one condition that cluster very differently than the other ones, and you maybe have other QC metrics that tell you that there might be a problem with that sample. That's an additional information, that this sample might be very wrong. And so hierarchical clustering or PCA or MDS plots are useful too. I mean, take the metrics, the metric of RPKM, and do a very classical hierarchical clustering. So, hierarchical clustering. So, I mean, I can.
Like how do the patterns of gene expression?
No. So here I mean at the overall expression. So not even looking at subset of genes.
Just think of a big heat map.
Big heat map.
Think of that between your two controls, the heat maps look similar, and your two treatments, the heat maps look similar.
Exactly.
Yeah.
Oh, okay.
Exactly.
Okay.
Exactly.
I was
That's it.
thinking.
Yeah. Just I think that's my last slide. Additional things that are possible to do with RNA-Seq. So as we described, we can do gene expression, we can do transcript expression, we can do alternative splicing, we can also do variant calling, and the most widely used tool for variant calling in exome and genome sequencing is called G8TK from the Broad Institute, and they released recently a best practice pipeline for RNA-Seq. And that could be used in conjunction with exome and genome sequencing, where you have a variant that you discovered in exome, and you can see the level of expression of your variant in the RNA-Seq. That could be useful to identify RNA editing events, all those kind of things. And the last one that I have to mention, because that's what I used recently and got a nice paper out of, is gene fusion discovery. And so fusion genes that can also be referred as chimeric genes and are very present in cancer can be detected in RNA-Seq data. And so there are tools for that, TOPHAT fusion, chimera scan, diffuse, snowshoes, and many others. And I have to make a plug for my personal best favorite. The one that changed my life is called Fusion Catcher. So that's the one we used to discover a fusion gene recently. And I think that's it for today. Yeah, references. Here are the website of some of the tools I described. I didn't put all of them, but I find a way to send you the missing links.
Oh, so the ones where the quality control, those are all
Yeah,
Picard?
so it's not, yeah, Picard and RCQC, and I'll send the link for that one.
Picard,
RCQC. RSEQC. RSEQC.
And we have the slides, right?
You have the slides, yeah.
We have all the slides, so.
Yep.
I got RSD.
No, RSE.
RSE.
RSEQC.
No, you're right.
RSEQC.
All right. Any other questions? Nicholas? All right.
Thanks.
Thank you.